Going over transcription in eukaryotes. Now, transcription follows three main steps. The first one is initiation, so how we are beginning to transcribe using the RNA polymerase. So first, it's essential to note that we have something called a eukaryotic promoter. And essentially what this does is a place for RNA polymerase to latch onto so it can begin transcribing. And so this promoter consists of a start point and in most eukaryotes, a Tata box. This Tata box can be seen in a diverse amount in diverse organisms in eukaryotes, and a lot of people argue that it's a common point of evolutionary basis. And so the start point, on the other hand, is a place where RNA polymerase actually begins to transcribe. So it doesn't begin over here, it begins at the start point. And next, it requires something called transcription factors, and in particular, there are many different types of transcription factors, but the ones that are extremely necessary are labeled as general transcription factors. And you may learn later on something called specific transcription factors, which will then regulate how transcription is done. But transcription factors are what is necessary in order for RNA polymerase to produce the RNA. And general transcription factors are present in every protein coding gene. It's absolutely essential. And only when the transcription factors are bound to the promoter is RNA polymerase 2 then able to bind to the promoter and then begin transcribing past the start point. Next, we're going to talk about elongation. This is pretty simple. It's almost exactly like DNA replication. It synthesizes the RNA nucleotides off of the 3' end, but there is one distinction, and that is that multiple RNA polymerases can act on the same DNA. And so especially when you notice that there are particular proteins that are necessary in the human body, in the cell, and so in order to keep synthesizing these proteins, multiple RNA polymerases would then have to synthesize the mRNA one after the other, after given a couple nucleobases difference. So they won't all do it at the same place. It will just be one will go first, and then after it's far enough, then the other one will latch onto the promoter and then synthesize. Last one is termination, and so in eukaryotes, it's a lot different from bacteria. So bacteria, once the start signal is reached, mRNA completely released immediately. But for eukaryotes, it takes a while, and it goes through something called a poly-A-dependent termination. So the poly-A signal is essentially the stop signal for eukaryotes. And so you would think that it would immediately release, like in bacteria. But nope. Once the RNA polymerase gets to the poly-A signal, it will actually transcribe the signal and continue down about 10 to 35 nucleotide bases past the signal until the mRNA is finally released. And so once it's released, it will cleave off the transcribed poly-A signal sequence, and it will go through some post-translation modification, commonly polyadenylation, which we'll go over right now. So kind of like what we talked about, it goes through something called polyadenylation, and so in general, post-transcription modification happens in two stages. The first stage is the addition of a 5' cap and a 3' poly-A tail. So this 5' cap is essentially just a modified guanine with 3 phosphates, and it just prevents degradation from the 5' end. The poly-A tail looks exactly like this. It's just a bunch of A nucleotide bases bound to each other, one after the other. It's a lot of them. So this is the poly-A tail, and both of these serve in order to not only protect the mRNA, but it allows them to exit the nucleus safely for translation to occur. And also, it can be involved in ribosomes being recognizing the mRNA so that it can be translated. Now, the second stage of post-transcription modification, is splicing. So you'll notice here that the mRNA is pretty long, but if you look at the actual mRNA that is going to be translated, it's much shorter. So it goes from here to here. That's because it is being spliced, the intron specifically. And a good way to remember what is being spliced out and what is being kept in is to note that introns are interfering. We don't want them. They're interfering with the mRNA, and so we get rid of them. On the other hand, the exons are the genes that are being expressed, so we want to keep them. And I guess another way you can think about it is X is exit, and the mRNA needs to exit the nucleus in order for it to be translated, so the exons will be kept in. So the introns sliced out, exons kept in, and so we have our coding segment now. And now you're wondering, what is UTR? UTR are untranslated regions. So they appear here and here. There are usually two of them, one at the 5' end and one at the 3' end. And so these are important as they do have some basis in gene regulation. And so even though they aren't actually translated or expressed, they still have meaningful contributions. And so splicing is done by a protein RNA complex called spliceosome, which, like I said before, is composed of proteins and small nuclear ribonucleic proteins, or small RNAs. And these small RNAs can also be called ribozymes because they act as an enzyme. Not only is spliceosome able to identify what segments need to be spliced out or spliced in, but it can actually catalyze its own splicing. And so we have ribozymes, and this is important as this is an example of an enzyme that is not a protein, it's an RNA. And so this is how we get splicing done. But that's just how post-transcription modification is done, and just a very quick overview of transcription in general.